Hi, my name is John Stella from the SUNY College of Environmental Science and Forestry in Syracuse, New York. I'd like to acknowledge my many co-authors. This work is a synthesis of 3 PhD students' research, Jared Williams and Melissa Rohde from SUNY ESF, and Chris Kibler from UC Santa Barbara, along with their mentors and other colleagues.
Riparian woodlands in dryland regions are hotspots for biodiversity, responsible for many critical ecosystem functions, such as mitigating high temperatures, creating habitat and wildlife corridors, and strengthening stream banks. Riparian ecosystems globally have been reduced and degraded by land conversion, channelization, flow regulation, and other interacting stressors. Decline of woodlands dominated by cottonwoods and poplars in the Populus genus are of special concern, as they are foundation species for dryland riparian systems throughout Western North America. The riparian woodlands are thought to be buffered against water stress by their landscape position and their favorable hydrology. Climate change, combined with groundwater extraction, increasingly threaten their long-term sustainability, particularly in drylands. Global trends toward hotter and drier conditions may contribute to groundwater declines that push these systems past a threshold of resilience. Numerous studies in recent decades document a range of responses, including reduced tree growth, canopy dieback, and in some areas, whole stand mortality. Over the last decade, California experienced its most extreme drought on record, with lower rainfall and snowpack and higher temperatures, resulting in soil moisture depletion and severe reductions in groundwater availability during the peak drought period from 2013 to 2016, when most of California was considered to be in severe, extreme, and/or exceptional drought conditions. During this period, groundwater levels declined statewide, in some cases dropping 30 meters or more over a few years. The effects of drought on woodland ecosystems can be observed in water stress indicators at different levels of resolution and from different vantage points. The challenge is to understand and then subsequently predict how trees and whole stands respond at the onset of drought, during its peak, and afterwards. At the individual tree level, we can look retrospectively at how growth and function responded using tree ring indicators, including annual radial growth and carbon stable isotope composition of the wood. Carbon isotope discrimination, shown here as delta 13C, provides a relative measure of water stress. Discrimination decreases with reduced transpiration induced by higher water stress. In this example, radial growth also declines because the reduced gas exchange limits photosynthesis. Though gas exchange usually recovers for surviving trees, growth may not if extensive branch and canopy dieback occurs. At a much broader scale, canopy level and river corridor scale level indices are observed from remote sensing platforms such as airplanes and satellites. With hyperspectral imagery, using many electromagnetic wavelengths, we can use mixing models to determine the proportion of a forest stand with green vegetation cover, or GV, versus non-photosynthetic vegetation, or NPV, which represents dead biomass. Canopy dieback events show as large reductions in GV and increases in NPV over time. We set out to understand the effects of the California drought on groundwater-dependent riparian woodlands. Specifically, what are reliable indicators of drought stress? How does severity of drought effects vary in space and time across indicators? And are there lags and thresholds relevant to ecosystem recovery and management? We were especially interested to understand which WSIs express water stress earliest and most dependably, and how they may inform ecosystem predictions and protection. We approached the questions using a range of scales and resolutions, from individual trees to landscapes, to compare indicators of water stress over similar timeframes. On the ground, we selected riparian woodland sites distributed along a climate gradient that varied in impact severity. We cored trees and measured their radial growth and carbon stable isotope ratios for the decade spanning the drought. We coupled the tree and stand level measurements with hyperspectral imagery from the NASA Hesperi airborne platform and analyzed live and dead vegetation change using spectral mixing models. At the broadest scale, we analyzed NDVI data, the Normalized Difference Vegetation Index, from the Sentinel-2 satellite platform to assess statewide patterns of canopy greenness during and immediately following the drought. Sentinel-2 resolution is finer than the older Landsat series, but was only launched in 2015. These complementary studies together allowed us to observe riparian responses to drought in space and time through different indicators. All three studies are published or in preparation. The Santa Clara River flows from the Mojave Desert to the Pacific Ocean in Ventura and Los Angeles counties in California. The basin has a Mediterranean climate with cool, wet winters and warm, dry summers, and many reaches of the Santa Clara are ephemeral. The river corridor has been subject to extensive urban and agricultural development over the last century, but the main stem of the river has not been severely altered or diverted, making it the largest river in Southern California that's mostly free-flowing. The native riparian woodlands along the Santa Clara are discontinuous, existing at locations called cienagas, where groundwater is close to the surface under normal conditions. Dominant species include two cottonwoods, Populus fremontii and Populus trichocarpa, as well as several willows. For the tree ring study, we cored 114 cottonwoods from 7 sites along a climate gradient throughout the river corridor and at sites that varied in drought severity impacts. For the remote sensing analysis, the hyperspectral imagery covered the entire river corridor, and we analyzed vegetation change at 6 focal sites that historically supported cienega forest stands. This top panel shows the progression of the drought as a time series of precipitation, which are the blue bars, and the Palmer Drought Severity Index, which is the red squiggly line, from 2010 to 2019, spanning the pre to post-drought periods. The gray shading indicates the peak drought years from 2013 to 2016. This middle panel shows radial growth of trees as the annual mean and standard error of the ring width index, or RWI, over the same period. Growth declined at a similar time and rate as PDSI, with the narrowest rings accrued during the peak drought. The recovery period showed high growth immediately after the peak, followed by 2 years with below average growth. The bottom panel shows the time series of delta 13C, or carbon isotope discrimination. Annual tree rings were analyzed separately for early wood when spring growth occurred and late wood corresponding to late summer. Reductions in discrimination in this water-limited environment reflect the drought stress response of trees closing their stomata to limit evapotranspiration. During the drought period, late wood discrimination declined earlier and was consistently lower than early wood, reflecting the effects of hotter and drier summers on leaf gas exchange. Groundwater decline induced drought stress in the riparian cottonwoods, with the strongest response as sites with the highest recession rates. The colored lines are monthly groundwater series from 7 sites, and the gray shading indicates the peak drought years. Groundwater depth varied across sites from 0 to 12 meters deep at the driest sites during the driest years. We calculated the rate of water table decline as the slope of the water level from the pre-drought years to their lowest point during the peak. Water table recession trends ranged from 0.1 to 1.75 meters per year, with some of the fastest declines in 2015 and 2016. Tree ring cellulose values of carbon isotope discrimination, delta 13C, shifted from their pre-drought baselines, the gray line, in site-specific patterns that matched the local groundwater dynamics. This is most apparent in the red line series from the Fillmore Cienega site, where tree function declined sharply in 2014 following a steepening of the groundwater recession rate. The greatest change in tree growth and function occurred at sites with recessions of greater than 0.5 meters per year. In addition, there was a depth threshold of approximately 4 to 5 meters that initiated reductions in growth and function. This threshold matches published values of maximum rooting depth for cottonwoods. In the second study, we examined 6 riparian woodland stands along the river, combining repeat flights of hyperspectral imagery with concurrent groundwater data to address changes in riparian woodland health. This map shows a portion of the corridor at the start of the drought. The river flows from right to left. There was a progressive brown wave of tree mortality characterized by decreases in healthy vegetation cover, or GV, in green, and increases in dead biomass, shown as NPV in red, which progressed downstream from east to west between 2013 and 2016. The blue regions are non-vegetated areas. The white arrows show areas experiencing notable dieback in particular years. Some of these areas, such as the eastern Fillmore-Cienega site and the central Sespe confluence, did not recover after the drought. We also found consistent, significant relationships between groundwater depth and GV, or healthy vegetation cover, and separately between groundwater and NPV cover, indicating that woodland health deteriorated in a predictable fashion as the water table declined at different sites and different times. Panel A shows the reduction in GV, and panel B shows the increase in NPV with increasing depth to groundwater. The effects become most severe beyond approximately 5 meters of depth, where the fraction of dead vegetation area surpasses that of live plant cover. This depth is similar to the 4 to 5 meter groundwater level threshold in the tree ring study that triggered declines in growth and leaf gas exchange. Based on these findings, the brown wave of vegetation dieback was likely caused by local changes in depth to groundwater associated with depleted riparian aquifers after years of extremely low precipitation and high summer temperatures. These factors suggest that semi-arid riparian woodlands are strongly dependent on shallow groundwater availability, which is in turn sensitive to climate forcing. We confirm these patterns more broadly in the statewide analysis of NDVI, another metric of vegetation greenness. Across California, NDVI and groundwater depth were coupled across a wide range of hydrologic regions. The negative trend lines in these figures show how NDVI varies with groundwater depth and how this varies seasonally. There was a strong coupling of NDVI with DTG in summer when water stress is greatest. There were similar trends for different woodland types, including those dominated by cottonwoods, willows, and riparian oaks. In locations with deeper groundwater, the lower NDVI value suggests a range of woodland responses, including reduced transpiration, photosynthesis and growth, greater canopy dieback, and cavitation-induced mortality. These relationships were strongest along natural streams, but significantly less along rivers and streams with altered flow regimes, where NDVI and DTG relationships were significantly weaker. In total, this statewide regional study suggests that patterns and processes observed in the finer scale river corridor studies are applicable to dryland biomes more broadly.
So some key findings from these complementary studies include that water stress indicators at vastly different scales confirm the groundwater dependence of dryland riparian woodlands, particularly during drought periods. Secondly, reductions in leaf gas exchange, growth, and canopy green vegetation followed groundwater level decline more than they did climate indicators.
There are numerous practical implications of this research, which are relevant to water managers in California and elsewhere. Numerous lines of evidence suggest that riparian woodland health declines when DTG is greater than 4 to 5 meters, or when water tables recede faster than 0.5 meters per year. Second, there are no clear prior warning signals across any indicators, no canaries in the coal mine. The effects on growth, leaf function, and canopy greenness occurred concurrent with and sometimes following the start of water table recession. In addition, many of these indicators would be logistically difficult and expensive to monitor in real time. Therefore, riparian monitoring programs should focus on high-frequency water depth measurements at monthly intervals or less. Groundwater depth is a master driver for many aspects of riparian woodland health. Many dryland regions lack adequate shallow monitoring well networks and robust data exchange between scientists and managers, so these should be prioritized wherever groundwater depletion is a threat.
Support for this work came from the NSF divisions of Geography and Spatial Sciences and Earth Sciences, as well as the U.S. Department of Defense CERTIP program. We're grateful to numerous colleagues who assisted with technical expertise, editorial suggestions, fieldwork, and property access. Thank you.